Hi, I'm Sarah, and my project is about comparing the cooperativity between various SOX transcription factors with Oct4, specifically in the context of induced pluripotent stem cells. So for background information, induced pluripotent stem cells are regular somatic cells that have been reprogrammed artificially to exhibit pluripotency, which is only naturally occurring in embryonic stem cells. So it's the ability to differentiate into any type of cell in the body, and this can be really useful in research and regenerative medicine. This reprogramming involves forcibly expressing 4 different transcription factors in the cell, which is Oct4, SOX2, KLF4, and MYC. In my project, I focus on the first 2, and as transcription factors, they target a specific DNA motif. And SOX and Oct can work together to bind cooperatively on SOX-Oct composite motifs. And in my project, there is the canonical motif and the compressed motif. The canonical motif is the main one that we want to focus on because this is what's present in the gene enhancers of thousands of genes related to pluripotency induction. Right now, the problem with trying to induce pluripotency is that there is a very low success rate with the wild type factors that I mentioned. So since then, it has been discovered that the cooperativity between SOX2 and Oct4 is a key aspect in achieving higher efficiency as well as higher quality of iPSCs. And that is what my project is focusing on, because a lot of labs have created mutants of SOX, not necessarily SOX2, but just a SOX in general that is capable of cooperating with Oct4 with a higher efficiency while still targeting the same motif, which is the canonical SOX-Oct motif. So this is where SOX17 comes in, because the mutants that I'm researching adopt features of SOX17 into SOX2 or vice versa. And this is because SOX17 has been found to bind Oct4 more tightly than SOX2. However, SOX17 and Oct4 do cooperate on slightly different motifs, mainly the compressed motif, which is more related to primitive endoderm development. So in this case, researching the compressed motif also functions as a control, because the compressed motif is related to a function that is antagonistic towards pluripotency induction. For the experiments that I had to do, I first had to express and purify the DNA binding domains of some of the SOX proteins, and once we have the purified proteins, we then mix them together with DNA under optimum binding conditions, and we let it sit for around an hour so that it has enough time to bind, and we do this at around 4 degrees. To visualize the different binding states, we use EMSA, which is a gel electrophoresis technique. It separates the different complexes by how it migrates through the gel. In this case, it's a native gel, and only the DNA is fluorescently labeled. So we can see that the bottom band is always going to be free DNA, and the larger the complex, the higher up it is, hence the shift. Finally, we quantify the band intensities, representing the relative concentration of the complex in the binding reaction, and from this we calculate the cooperativity. Here we can see that SOX17 FNV is the only mutant that has a significantly higher cooperativity with Oct4 relative to SOX2, specifically on the canonical motif. I also got to do structural modeling of the heterodimer interface, and here we can see that SOX17 FNV has more hydrophobic residues. More specifically, the switch from alanine to valine at position 61 allows the isoleucine in Oct4 to be more engaged. So to conclude, SOX17 FNV is the strongest candidate for iPSC induction based off of its cooperativity with Oct4, and this is possibly explained by the stronger hydrophobic and van der Waals interaction at the heterodimer interface. So that will be everything about my project. If you have any questions, please don't hesitate to email me, and thank you so much for listening.